All living organisms utilize a variety of coordination complexes called hemes to help with things like oxygen transport, oxygen storage, and electron transfer in the body. These hemes have to be built, and this process involves many enzymes combining substrates to assemble these structures. Without heme formation, processes like oxygen transport, oxygen storage, and electron transfer will cease. In the biosynthesis of hemes, the second step of assembly is catalyzed by the enzyme delta-aminolevulinic acid dehydratase, or ALAD for short. ALAD is made up of 8 identical subunits. In each subunit, there is a metal center, zinc. Zinc is able to bind to ALAD because ALAD is a tridenate chelator, which uses 3 ligands that act like a claw to bind tightly to zinc and hold it in place. These 3 ligands are cysteine residues. The zinc will coordinate with the thiolate from each cysteine, and that will help hold it in place. The sulfurs bind to zinc because of hard-soft acid-base theory, where acids and bases are classified based on their charge to radius ratio, and acids and bases with similar ratios preferentially pair with each other. Sulfurs are classified as soft bases, so they will seek to pair with soft acids. Zinc is an intermediate acid, so it would be able to interact with either hard or soft bases. It is on the softer side of intermediate acids, so the interactions with sulfur will still be favorable. It is also important to note that zinc is a D10 metal. D10 metals can prefer either square planar geometries or tetrahedral geometries. In this case, the tetrahedral geometry has less steric strain than the square planar geometry, and so, therefore, the tetrahedral geometry will be preferred. The tetrahedral geometry, along with zinc being a D10 metal, means that when the ligand field stabilization energy is calculated, thermodynamically speaking, zinc is expected to be labile. This is because there is no ligand field stabilization energy. However, there are strong electrostatic interactions with the partial charges that bind to zinc. These electrostatic interactions work to help stabilize zinc, explaining why zinc is inert when bound to ALAD. Since there are 10 d electrons in zinc 2+, each ligand will also contribute 2 electrons to the total electron count. This is a total of 16 electrons. Zinc is normally bound to either water or the substrate, which will contribute another 2 electrons to the total electron count. When there are 18 electrons in the total electron count, zinc is coordinatively saturated and stable. Without zinc, ALAD would not be able to function. While the exact role of zinc is unknown, we know that it is important in helping aid the combination of 2 molecules of 5-aminolevulinic acid, or ALA, to form porphyrbilinogen, or PBG. There are 2 proposed mechanisms for this combination process. Both of these mechanisms involve an A and a P site. In the A site, zinc coordinates with the nitrogen on the N-terminus of ALA. The other ALA molecule coordinates at the P site. They are then combined to produce the PGB molecule. From here, PBG will go through a process which will further transform it into a heme. This may seem as if zinc is only a structural scaffold, but its role goes beyond that. One proposal is that zinc acts as a Lewis acid and then later acts as a base. It may also stabilize intermediates of the catalytic reaction. Without zinc, ALAD does not combine ALA molecules into PGB molecules. This halts the production of hemes. One example of when this happens is when lead inhibits the binding of zinc to ALAD. Lead will inhibit the binding of zinc to ALAD because lead is also an intermediate acid, but even softer than zinc. Although there are no major conformational changes when lead binds, lead will not act as a Lewis acid and then as a Lewis base to catalyze the reaction, and it will not stabilize reaction intermediates, which prevents PGB formation. Hemes, which are assembled in part from PGBs, cannot be formed without ALAD. Halting the production of hemes can cause serious side effects like neurovisceral attacks, nausea, abdominal pain, cognitive delay, and many more. ALAD is found in every living organism. Without it, heme production would not be possible. So the next time you see something living, moving, and working, remember the important role that ALAD plays in getting there.